Welcome to the latest installment of the AST Journal Club series. Today's session features a presentation and discussion on indirect CD4 positive T-cell epitopes associated with transplant rejection and targets for donor-specific tolerance induction. This session is sponsored by AST's community of transplant scientists. In a moment, I will turn the session over to our moderator, Dr. Paolo Cravedi, from the Icahn School of Medicine at Mount Sinai in New York. But before we begin the main presentation, we do have a few housekeeping notes to help you engage with today's session. There is currently a viewership polling question displayed for the audience. Please take a moment to answer this question while we finish the remaining announcements. We encourage you to use chat during the Journal Club to share your thoughts and comments. However, if you have a question to submit to our panelists, we recommend using the Q&A button in your Zoom webinar panel instead. Questions submitted via the chat window may be missed during the presentation. This Journal Club is being recorded, and the archive will be available on the MyAST website and the AST YouTube channel in 2 to 3 business days. Finally, when you log off at the conclusion of today's Journal Club, you will see a link to a short evaluation survey to complete. Please fill out the survey to help us keep our content current and engaging. I will now turn the session over to our moderator, Dr. Cravedi, to begin the presentation.
Thank you, Brian, and good afternoon, everybody. I'm very excited to introduce you our panelists today. We have Zhuldyz Zhanzak, who's going to be our speaker that will walk us through the results of this important study. And then we have two discussants, Haydn, Kissick, and Chris Larsen from Emory. Zhuldyz is a PhD student at Emory Transplant Center under the supervision of, again, Chris Larsen and Haydn Kissick. And her work is focusing on the identification of the TCR specificities of CD4 T cells in mice and human transplant recipients. So this is a very important, hard to study, probably understudied topic. And now I just want to turn over to Zhuldyz and leave the question for the discussion.
So thank you very much for this nice introduction, and we appreciate this opportunity to present our recent research on identifying the indirect CD4 T cell epitopes and the ways how we can target them for donor-specific tolerance induction in transplantation. So for the conflict of interest, we do not have any relevant financial relationships to disclose. So to start off, transplantation is a life-saving treatment for patients with terminal organ failure. And even though the short-term outcomes post-transplantation are good, the long-term outcomes are suboptimal due to the T-cell-mediated and the antibody-mediated rejection. And unfortunately, in most of the cases, patients post-transplantation heavily rely on the toxic immunosuppressive drugs that are in most of the cases are generic. So from immunological perspective, rejection is a complex immune process with multiple effector mechanisms. So what happens is after the transplantation, the donor antigens gets presented to antigen-presenting cells, which processes them and presents to CD3 positive T cells. And once activated, these CD3 positive T cells become CD8 T cells, which later on contribute to the T cell mediated rejection. Whereas another part of these CD3 positive T cells become CD4 T cells, and a part of them provide help to B cells in the production of the antibodies. And these antibodies later on contribute to the antibody-mediated rejection, which is the main focus of this work. So the mechanism of how these antibodies are made in transplantation is called indirect recognition. So what happens here is that donor major and the minor histocompatibility antigens labeled in red gets presented to the recipient MHC class II expressing antigen presenting cells shown in gray. And this APCs later on processes donor antigen and presents them to recipient CD4 T cells. And the recipient CD4 T cells that have been activated through this pathway are called indirect CD4 T cells. And one part of these indirect CD4 T cells provide help to CD8 T cells in the production of interferon gamma, whereas another part of these indirect CD4 T cells differentiate to Tregs, Th17s, Th1s, or T follicular helper cells. And particularly these T follicular helper cells later on provide intramolecular help to to B cells in the formation of antibodies. And it is well known in the field that these antibodies drive the transplant failure. So in our previous study with 594 kidney transplant recipients, we used this algorithm called PURGE, which calculates the number of potential indirect CD4 T cell epitopes generated from a donor to recipient. And we used it to stratify the patients. And we found that the kidney transplant recipients with a higher number of indirect CD4 T cells, reflected by a high PURGE score shown in red line, undergo a high rate of the rejection and the development of the antibodies. In contrast, patients with a fewer number of indirect CD4 T cells, those that have a low PURGE score and those that have a black line, undergo a lower rate of the rejection and the development of antibodies, which helped us to generate a hypothesis that these indirect CD4 T cells are bad for transplant outcomes. However, even so, it is well known the gap in the transplant field is that the paucity of these well-defined indirect CD4 T cell epitopes has hindered the development of antigen-specific tolerance strategies. And in this work, we are trying to address this gap in the field by identifying indirect CD4 T cell epitopes in transplant patients in a murine model of allograft rejection. And finally, we are aiming to target them to achieve the tolerance induction. So throughout this presentation, I will be covering this step by step, and the first we'll start off how we identify these indirect CD4 T cell epitopes in transplant patients. So when we started this work like 5 years ago, this paper had just been published showing that heart transplant recipients with particularly HLA-DQ antibodies reflected by black line have a high rate of the rejection compared to patients that have non-HLA-DQ antibodies shown in gray. And the majority of the antibodies that were made in these heart transplant patients were against the HLA-DQ, like you can see in green bar graph, which made us to generate this hypothesis that perhaps donor HLA-DQ is a pathogenic antigen. So around this hypothesis, we asked this question about what the donor HLA-DQ antigens are recognized by indirect CD4 T cells in transplant patients with HLA-DQ antibodies. So to address this question, we utilized a prediction system in which we used an algorithm called Epitope R, which was previously developed by our lab, and it is based on publicly available algorithms such as NetMHC2PAN and the IEDB. So with this prediction, we essentially predicted 15 mer recipient HLA-DRB1 binding class 2 peptides that are derived from the donor HLA-DQ alpha and the donor HLA-DQ beta, but those that are different from the recipient's self-DQ alpha and the DQ beta. And to demonstrate how this prediction works is that when we align the donor and recipient HLA, not every region is the same. There are some portions of the HLA which are the same, and the more likely they do not elicit an immune response. But there are also a high proportion of these HLAs that consist of the polymorphic regions and mismatched amino acids, and those are the regions that more likely contribute to rejection. And what this prediction does, it tries to focus on these polymorphic regions and it tries to predict the peptides from them. And this pipeline not only works for transplantation, but it's also translational in autoimmunity for self-antigens, in cancer for neoantigens, and for infectious diseases for viral antigen identification. So with this pipeline, we predicted and synthesized close to thousands of peptides. And we validated it by recruiting kidney and heart transplant recipients with HLA-DQ DSA, without HLA-DQ DSA, and healthy individuals that we use as an active control. And we tested We tested these peptide pools by performing this T cell expansion for 2 weeks with these predicted peptide pools together with a low dose of IL-2 and IL-7. And as a readout, we measured the T cell response against these predicted peptide pools. So the first assay that we used was interferon gamma ELISpot, which is essentially an assay that measures the T cells that produce interferon gamma in response to a specific antigen. So we found that upon stimulation with these predicted peptide pools, patients with HLA-DQ antibodies have a stronger T cell response in comparison to patients without HLA-DQ antibodies and the healthy individuals. We also validated it by intracellular cytokine staining assay, in which we measured the cytokines such as TNF-alpha and the interferon gamma produced by CD4 T cells. And we found that patients with HLA-DQ antibodies secrete these TNF-alpha and the interferon gamma cytokines more robustly compared to patients without antibodies. So after validating that these peptide pools elicit an immune response, we wanted to identify the select immunogenic peptides derived from these peptide pools that cause a response. And for that, we performed the single epitope mapping, which is essentially a method to identify a T cell response to a given peptide by measuring the T cell response to each of them. So for that, we utilized this interferon gamma ELISpot assay again. So this graph can be a bit heavy, but essentially what it shows, the y-axis is a magnitude of the T cell response, and the x-axis demonstrates the amino acid position of the peptides that have been mapped. The top graph demonstrates the peptides that have been predicted from the HLA-DQ beta, whereas the bottom graph demonstrates peptides that have been predicted from the HLA-DQ alpha. So with this idea, assay, we found two regions called as region 1 and the region 2 that consistently elicits a T cell response in multiple individuals. And here each line demonstrates one patient uniquely labeled with a color. So more specifically, region 1 covers about 4 peptides from about 9 to 21 amino acid position of HLA-DQ beta, and the region 2 consists of about 20 peptides derived from about 60 to 110 amino acid position of HLA-DQ alpha. And more specifically, the region 2 peptides are correlated with the abundance of HLA-DQ antibodies in these patients. So after identifying these immunogenic HLA-DQ peptides, we asked this next question about whether these select immunogenic HLA-DQ antigens can be used to identify HLA-DQ specific TCRs in transplant patients. And to address this question, we developed this assay in which we generated these artificial APCs, which are essentially just synthetic cell lines that express a patient-specific HLA-DRB1 as an HLA allele and the CD86 for co-stimulation. And we co-cultured each of these patient-specific HLA expressing APCs with CTV cell trace violet labeled CD4 T cells. And we found that upon stimulation with specifically HLA-DQ peptides that we have previously mapped, these cells upregulate CD25, which is a T cell activation marker, and dilute the cell trace violet, which is a proliferation dye. Meaning that these cells respond to these HLA-DQ peptides. And we don't see this response when we stimulate them with irrelevant HAV peptides. Later on, we sorted out the CD25 high CTV diluted CD4 T cells and performed single cell TCR sequencing to identify clonally expanded TCRs, which we later on expressed in the TCR alpha beta knockout Zhuldyz cell lines. So with this analysis, we got 4 paired TCR alpha and the beta clonally expanded TCRs, and we showed that upon expressing them into this TCR alpha beta knockout Zhuldyz, they can upregulate these T cell markers such as CD3 and the TCR alpha beta. And however, we wanted to further test whether these TCRs are functional. Therefore, we adjusted our assays that we previously showed. So this time, rather than co-culturing these single HLA expressing artificial APCs with bulk CD4 T cells, this time we are co-culturing them with TCR expressing Zhuldyz. So this is pretty sensitive assay between a single HLA expressing APC and a single TCR expressing Zhuldyz. And we found that upon stimulation, these cells with HLA-DQ peptide, these cells secrete cytokines such as TNF-alpha. And however, this response does not occur when we stimulate them with irrelevant influenza peptide, which shows that not only we can find these HLA-DQ specific TCRs from transplant patients, but also can show that they are functional. So to wrap up this part one of our work, about identifying these indirect CD4 T cell epitopes, we show that by utilizing this prediction system, we identified that HLA-DQ antigens can elicit a robust CD4 T cell response in patients with HLA-DQ antibodies, and that these patients have CD4 T cells specific for two polymorphic hotspots, and that select HLA-DQ antigens can facilitate the identification of HLA-DQ specific TCRs. So this human work helped us to build the foundation and get some preliminary data. However, we wanted to dig more into the mechanism of indirect recognition, and since the human work has a limitation due to the limited number of patient samples that we can recruit in our study, we shifted to a murine mouse model to ask more fundamental questions. So before we'll show any data, we just wanted to give some introduction about the mouse models that we use in the lab. So at Emory Transplant Center, we do this full MHC mismatch and the single MHC mismatched allogeneic transplant models. So for the full MHC mismatched model, we perform skin graft transplants from the donor biopsy into the recipient block 6, in which every class 1 and the class 2 antigens between this donor and the recipient are different. We also perform the single MHC mismatched transplant models between the donor block 6 KD and the recipient block 6, in which the only foreign antigen is a class 1 antigen called KD. And as an active control, we perform syngeneic skin grafts from the block 6 to block 6, in which every antigen is self. So in every of these models, we perform skin graft transplants. We wait for about 2 weeks until the peak of the rejection occurs, and then we sacrifice the mice and then mostly look at the draining lymph node and the spleen, where the strongest T cell responses are. So with this model, the very first question that we asked was, which biopsy MHC antigens generate a strong antibody response in block 6 mice? So to address this question, we performed this experiment in which we did skin graft transplant from the biopsy mouse into the block 6. We kept the mice around for about 5 weeks, bleeding them every 7 days, and we measured the antibody response. We found that as early as day 14, the antibody response to class 1 antigen called KD was the strongest compared to other antigens, as you can see from this red line, which made us to have this hypothesis that perhaps KD antigen elicits a potent antibody response. So around this hypothesis, we asked this next question about what donor H2KD antigens are recognized by indirect CD4 T cells. And to address this question, we again used this prediction pipeline called Epitope R that we previously developed by Larsen lab. So essentially, in this mouse model, we tried to predict 15-mer recipient IAB binding class 2 peptides that are derived from the class 1 and the class 2 antigens of the donor biopsy, but those that are different from the recipient's class 1 and the class 2 antigens. And we tested, we predicted these peptides, we synthesized them into first peptide pools based on the MHC alloantigen type, and we measured the T cell response by interferon gamma ELISpot. And we found that the T cell response to this class 1 antigen called KD was the strongest in this rejecting allogeneic group shown in red compared to the non-rejecting syngeneic group, which made us to think that probably KD is an immunogenic antigen. That's why we see a T cell response against them. However, we further wanted to identify select immunogenic antigens within this KD peptide pool that elicits an immune response. And for that, we performed this single epitope mapping assay in which we measured the T cell response in which we measured the T cells that secrete these interferon gamma cytokines in response to the select KD antigens. And we found 4 immunogenic peptides that elicits an immune response above average. And those peptides are labeled in red. And when we try to align the sequence of these immunogenic peptides with the self sequence, we see only 2 amino acid mismatches that are highlighted in red. And particularly for this study, we'll be focusing on this antigen called KD287, which is labeled in red. So essentially, this is a CD4 T cell epitope that maps around around 287 and the 301 position of this KD protein. So, and what this alignment demonstrates is that these 2 amino acid mismatches are somewhat sufficient to make this epitope to be rejection associated. But then after identifying this immunogenic peptide, we wanted to be able to track these donor-specific T cells. And the NH tetramer core was very helpful for us to generate tetramer positive CD4 T cells, and we generated this double color APC and PE conjugated tetramer positive CD4 T cells from the draining lymph node of this rejecting allogeneic group. And we found that we cannot find these donor-specific CD4 T cells in the draining lymph node of non-rejecting mice. And on average, we can find about 0.2% of tetramer positive CD4 T cells. And upon phenotypic characterization of these tetramer positive CD4 T cells, we can see that these donor-specific T cells are activated, upregulating T cell activation markers such as PD-1 and the CD44. However, non-tetramer population, bulk, which are essentially the bulk CD4 T cells, they're not as highly activated. Further, we wanted to understand the fate of these donor-specific CD4 T cells. And for that, we looked at the 3 main CD4 T cell subsets, such as Th1s, Tregs, and the T follicular helper cells. And for Th1s, we look at the transcription factor Tbet. And for Tregs, we look at the FOXP3. And for T follicular helper cells, we look at the surface marker CXCR5. And the analysis that we performed was essentially we compared tetramer versus non-tetramer CD4 T cell populations. And we didn't see much difference in the expression of the Tbet, like you can see from these representative flow plots, and there was no much difference in the expression of the FOXP3, like you can see from this top quadrant. However, the only difference that we saw was in the expression of the CXCR5, which is a surface marker for T follicular helper cells. And to reiterate, the T follicular helper cells are the subsets of the CD4 T cells that particularly provide intramolecular help to B cells in the formation of antibodies. So this phenotype was pretty captivating to us and made us to ask this next question about the functional role of these indirect KD287 CD4 T cells. And to address this question, we performed this adoptive transfer experiment in which we first did this skin graft transplant from the donor B6KD into the recipient B6 mice. To reiterate, the only foreign antigen here is a class 1 antigen called KD. So with this model, we performed skin graft transplants. We waited for about 2 weeks until the rejection occurs, and we validated that this model results in the rejection and the development of antibodies. At the same time, we performed this model in which we did skin graft transplants from the donor B6KD into the recipient TCR alpha beta knockout mice. And after 2 weeks post-transplant, we validated that this group does not undergo rejection, and there was no development of the antibodies. So after finding out that one group rejects and another group does not reject, we flow sorted KD287 CD4 T cells from this rejecting group and adoptively transferred them into this non-rejecting skin grafted TCR alpha beta knockout mice. And each mouse received for about 20,000 tetramer positive CD4 T cells. And as an active control, we transferred the same number of the LCMV specific SMARTA CD4 T cells that we use as an active control. So after the transfer, we waited for about a month to see whether this the recipients would undergo any difference. So we noticed that as early as day 10, the mice that received these KD287 CD4 T cell tetramers shown in red start to develop a KD-specific antibody response. However, the mice that received this LCMV-specific CD4 T cells, they don't develop these KD-specific antibodies, which shows the specificity of the transfer. We also noticed that before the transfer, the skin of these grafted mice were completely intact. On the day 11 post-transfer, the inflammation started to occur. So by day 15, it progressed, and by day 17, all the grafts were rejected. And as a histological analysis of the skin of the naive black 6 mice demonstrated a low concentration of the infiltrating cells, whereas the skin of the mice that underwent this adoptive transfer had a high concentration of the infiltrating cells. Which indicates that the transfer of these KD287 CD4 T cells may contribute to allograft rejection. So to summarize this second part of our work here about the identifying the CD4 T cell epitopes in a murine model of allograft rejection, we showed that again by utilizing this prediction system, we found that donor H2KD antigens can elicit a potent antibody response and a CD4 T cell response, and that this rejection-associated KD287 CD4 T cells can contribute to antibody formation and the rejection. So in the last part of our talk, we'd like to explain how we are thinking to target these indirect CD4 T cell epitopes for tolerance induction. So the main questions that we are trying to answer in this last part is, can these indirect CD4 T cell epitopes be targeted to induce donor-specific CD4 T cell tolerance? So we spent a fair amount of time thinking how we can answer this question. And we backed up a bit and went to the fundamental T cell immunology. So we know that the classical T cell activation requires 3 main signals. The signal 1 between peptide MHC and the TCR. The signal 2 is a co-stimulation such as CD28, such as an interaction between CD28 and the CD886, and the signal 3 is a cytokine. And we thought that perhaps by manipulating 1 or 2 of these signals, we can achieve the T cell tolerance. And for the signal 1, we are trying. So we thought that that we can manipulate the signal 1 by administering this high-dose H2KD peptide. And the rationale here is that by loading these antigen-presenting cells with so much antigen, the T cell signal strength will be so strong that it can be overloaded by the dose of the antigen. And for the signal 2, we decided to administer the co-stimulation blockade called the CTLA-4 which is a fusion protein that inhibits T cell activation by blocking the interaction between CD28 receptor. And it is clinically available and previously developed by our lab. So with this idea, the mouse model that we came with was that we take these naive black 6 mice, we administer systemically a high dose of these H2KD peptide pools, and then on day 1, we treat it with a co-stimulation blockade called CTLA-4 and then we deplete the CD8 T cells so that we'll be only targeting the CD4 T cell tolerance. And then on day, and to note, all these treatments were performed under non-inflammatory conditions. And then after these treatments, we bring the challenge in the form of the skin graft transplant from the B6KD donor, in which the only donor foreign antigen is a KD. So it is single MHC mismatched transplant model. So we perform these transplants, and then we keep depleting CD8 T cells so that they do not come into the circulation. And we kept the mice around for about 2 months to see what happens. And we found that the mice that underwent the rejection, as shown in green, they have elevated antibody response. However, the mice that received this treatment, that I will be calling as peptide treated for simplicity, they had a reduced amount of the antibody response against the donor KD antigen. And interestingly, the mice that received this peptide treatment had a prolonged graft survival in comparison to mice that underwent the rejection. We further wanted to understand the cellular what is happening on the cellular level. Therefore, we looked at these donor-specific CD4 T cells, and we found that the mice that underwent the rejection, even after 2 months post-transplant, they have these tetramer positive CD4 T cells, whereas the mice that received this peptide treatment are not, there are not many donor-specific CD4 T cells left. Like you can see that the both frequency and the number of these donor-specific CD4 T cells reduced in the mice that received this peptide treatment. And the phenotypic characterization of these cells demonstrate that mice that underwent the rejection are still pretty active, upregulating T cell activation markers such as CD4 and the PD-1. However, the mice that received this peptide treatment had a very few number of the CD4 T cells, like you can see from these representative flow plots. We then wanted to understand if so this phenotype this data was very interesting to us, and we asked why it's happening, why these mice that received peptide treatment had such a few number of the CD4 T cells left. Therefore, we decided to do this TCR sequencing to look at the repertoire of these mice. And for that, we did this experiment in which we first performed skin graft transplants from the B6KD to BLAK6, and 2 weeks after the rejection, we flow sorted KD287 CD4 T cells and performed TCR sequencing. At the same time, we did these treatments with peptides, CTLA-4 Ig, and the CD8 depletion. And we also did the skin graft transplants, and after 2 weeks post-transplant, similarly, we flow sorted tetramer positive CD4 T cells and did TCR sequencing. To avoid being biased, we downsampled to the lowest number of the clonotypes, so that we are working with an even number of the clonotypes. And the first readout that we looked at was TCR diversity, and we found that the mice that underwent this peptide treatment, even after downsampling, they have a very narrow TCR diversity. And the repertoire of the mice that underwent this rejection, they was very polyclonal, consisting of very small and the medium-sized clonotypes, whereas the repertoire of the mice that received peptide treatment were very oligoclonal, consisting of the large and the hyper-expanded clonotypes. So this data made us to generate this preliminary hypothesis that we still don't have a very solid answer, that perhaps some of the alloreactive TCR clones are missing. So to support our hypothesis, we tried to track these alloreactive TCRs that we previously identified from our work, but due to the time limitations, we are not able to present it today. But essentially, when tracking these public KD287 specific TCRs, we can find that under the condition of the rejection, there are a high number and the frequency of these public TCRs. And similarly, when we try to track this by a similar quasi-public TCRs, we can find the high frequency and the higher number of these quasi-public TCRs under the condition of the rejection compared to the conditions of the peptide treatment. So to summarize this last part is that we think that this combination treatment of high-dose H2KB peptide pools with this co-stimulation blockade called CTLA-4 Ig can mitigate the donor-specific CD4 T cell immune response by pruning the TCR repertoire and creating the holes in it. So to summarize our work, we started our presentation with this gap that the paucity of the well-defined indirect CD4 T cell epitopes has hindered the development of antigen-specific tolerance induction, and we are trying to address that gap by utilizing this prediction system using this algorithm called epitope R, which is applicable both for the human systems and the mouse systems, and we demonstrated that in the human work that kidney and the heart transplant recipients have two polymorphic hotspots within the HLA-DQ that consistently elicit immune response in multiple individuals. And in a mouse system, we identified an CD4 T cell epitope called KD287 that can contribute to rejection. And mechanistically, we showed that indirect CD4 T cells, under the condition of rejection, are highly activated. They maintain public TCR repertoire, and they have these robust donor-specific antibodies. However, we can manipulate these indirect CD4 T cells by doing this high-dose peptide immunotherapy with co-stimulation blockade CTLA-4 Ig, and eventually we can achieve the reduced frequency of these indirect CD4 T cells and the pruned TCR repertoire, so that eventually we can have this reduced donor-specific antibodies. So for the translational significance and the next steps, we think that by targeting this, so our current working hypothesis is that by targeting these few indirect CD4 T cell epitopes before the transplantation can mitigate this immune response in mouse, then perhaps by targeting these hotspots, these polymorphic hotspots within this HLA-DQ antigen, before the transplantation, we can achieve this promising strategy towards personalized medicine. And I guess the future direction would be to do this more fine mapping of HLA-DQ antigens, and with enough efforts, we can do this HLA-DQ peptide therapy to prevent HLA-DQ donor-specific antibodies in patients. So with that, I would like to acknowledge my PIs, Dr. Larsen and Dr. Kissick, and everyone in Larsen lab and in Kissick lab, Boss lab, Rob Fairchild for giving us B6KD mice, the clinicians who helped us to recruit patient samples, Emory Genomics Core, NIH Tetramer Core, Emory Flow Core, and the funding agency. And with that, we'd like to, we'd be happy to get the questions and open for discussion. Thank you.
Thank you, Zhuldyz. This was a very elegant paper, a tour de force. You could see the amount of work behind all these very elegant experiments. So now I'd like to open the discussion. As I mentioned at the beginning, we have two discussants. We have Haydn Kissick, who's an assistant professor in the Department of Urology at Emory. His work focuses on understanding the basic mechanisms regulating T-cell immunity to cancer, viral infection, and transplanted organs. And the second discussant is Chris Larsen, who is professor of surgery in the Division of Transplant at Emory. As probably you all know, Chris Larsen is the father of co-stimulation blockade, and his studies form the basis for the development of CTLA-4 belatacept that is currently used in the clinic. And more recently, as we just heard, he's been working on TCR sequencing, and he developed pipelines to predict the immunogenic peptides. So we already have quite a few questions. There's a few questions that are focused on the other pathways, the direct presentation. How do you see a role of direct T-cell mediated recognition, or do you think that indirect is dominant? I would say dominant also over the semi-direct presentation.
Yeah, I'm happy to start with that. So this work really focuses on the indirect pathway because of its role in the development of donor-specific antibodies. And we know that's a really important pathway for late graft failure. This is not to say that direct recognition is not important. In fact, we think it's quite important. It might be important early. There's lots of data that suggests that direct recognition can be sufficient, and it's probably helped by indirect recognition later. And so, we don't want to overstate that when we think about this as a tolerance strategy, it's to tolerize this arm, which is an important independent arm. So we do think the direct pathway is important and of great interest.
And it's something the second we had a paper at the same time that was did we lose everyone?
No, just click that button over there just for a second. Can you guys still hear us?
We
We
can
can
still
hear
hear
you.
you. So
Yes. Okay, we'll just talk. I mean, we almost at the same time there's a paper on direct recognition. This absolutely contributes.
There's a lot of question from Neil that is asking whether in the mouse model there's a dominance of responding cells. Are they naive or memory?
I can probably answer that the best. They're not memory. So we don't think of a memory cell. When there's antigen present, it's probably not a memory cell because it's responding to the antigen. But then they're very diverse. So there's definitely something very close to a TFH cell, CXCR5 positive. There are definitely TBET, so TH1 canonical TBET positive cells within that tetramer pool. And then it's heterogeneous within that. There's many other populations. There's been lots of work, in the last few years by lots of different groups, including Transplant, on what they call stem CD4 cells, which is sort of close to a memory cell, undifferentiated. It has potential to divide and do things. Looks like there's cells like that within the tetramer pool. So that CD4 pool is, I would say it's very specialized in many different roles. And I think understanding exactly what those cells are doing is really important. And I think what the tolerization regime is doing to change that is that's what I find the most interesting about this, actually.
Yeah. And just, another thought that may be behind that question is whether or not there's heterologous immunity, so prior memory responses to pathogens, are they contributing? This model really doesn't address that. These are naive mice. And in contrast to, say, direct recognition, where I think we know the frequency is really high, maybe a % recognizing a particular allele, based on other work by Mark Jenkins, and I think Zhuldyz work gets at it, indirect epitopes are probably very, very low frequency. There may be only 500 KD287 specific naive cells in an unsensitized mouse. So yet there could be cross-reactivity, but just stochastically, it's probably less important for indirect to have heterologous than it is for direct, but we don't directly assess that in these studies.
I'm not sure a mouse could ever really answer that.
Yeah.
That particular, if it was, like it was a cross-reactive memory cell to a different, I don't think the mouse is the right place to do that.
There's you partially already addressed that, but there's a question about the fact whether you think that pre-sensitized mice would respond similarly to the proto-allergenic strategy to CTLA-4 plus the peptides. And maybe as a segue, how do you think this could be translated as a tolerance approach in the clinic? Yeah.
I can answer the first part, and the second part I can answer. So we actually did this experiment in which we compared normal rejecting mice and the mice that first underwent the rejection, and we started the treatment, I think, day 14 post-transplant. And we didn't see much difference in the development of the antibodies. So I think that this treatment probably does not work in the condition when there are already like memory T cells and the memory B cells. So that was the first question.
And I'll just add, because I think Chris will answer the clinical question the best, but Chris and I spoke about exactly this morning. Once you have a DSA response, all the CD4 help probably drove that, to make plasma cells and to make, affinity matured B cells that are making anti-graft antibodies. But once that's happened, the CD4s might be completely dispensable. Like you've lost the battle, the process has happened. So I do think what we're talking about here is a pre-sensitizing to a narrow range of epitopes that could prevent DSA formation in the future.
Yeah, one other thought on that topic is there's great work going on from Duke, UCSF of trying to desensitize patients who have broadly HLA-specific antibodies, and they use co-stimulation blockade at the same time. And so this kind of peptide treatment, is not going to kill a memory, the memory CD4 may not be important if the plasma cells are still there, but if you kill the plasma cells, and we're getting, sorry about the extras, it still could potentially be a part of it. But probably just CD28 blockade plus peptide is not going to kill a memory cell. So translation, yeah, this is a beginning point. From a person that answered in 1996, how long would it take to get CTLA-4 IG in the clinic? And I answered, I don't know, 5 or 6 years, and 20 years later, it wasn't there. So don't listen to what I say about how long it takes. But I think, again, this is the basis for understanding maybe a point of leverage in preventing. If you have a heart transplant and you develop a DQ-DSA, it's going to shorten your life. And so if we can understand what the help is behind that DSA, the DQ-DSA, then I think it's a leverage point. So it's going to take a lot to translate this. It probably won't be peptide pools. It might be mRNA. It might be other ways to express the antigen, but it's a starting point.
And what I was excited about from that result with the hotspots is it's only very narrow pieces of the donor antigen. You don't need to work out every patient, every part of every patient's one. So it always seems to be the same. So it just narrows the challenge that antigen-specific tolerance is. It seems so insurmountable when you think about it, but it's not as bad as we maybe first thought. And maybe if we start looking, we didn't look at DR, response to DR. If we start looking at that, and it's also just a narrow range, and it's always the same little bits, maybe all of a sudden it's not that many antigens that you have to just bring down. And like Chris said at the start, we don't think that this is tolerance. This is one arm of the immune response to a graft. We might bring it down 25%, and then patients who are on the edge, that was the tick over the bring their immunity down and helps them.
So this would be probably more like an add-on kind of treatment. That's the way you envision that.
Yeah, I mean, if you think about it on top of current therapy, yes. So maybe you can even do it post-transplant. If you have stable patients who are at risk for DSA on pelitacicept, maybe you could just give peptides. Or you could think of it as a component of a multimodal approach for tolerance. So if there's a different approach to get tolerance to the CD8 pool, this plus that, starts to really diminish the immune response. So I think of it either way, as an add-on to current therapy or as one piece of a multimodal tolerance approach.
And let me try to clarify one point. You mentioned you have this epitope R pipeline that is gonna predict the peptide, the sequences of the peptides, knowing the mismatch between the HLA-DR. Do you still need to test which ones are the peptides in those specific patients, or based on this prediction, you would just give those peptides?
Yeah, you want me to do that one?
I mean, how good is this?
Why don't I let Zhuldyz start, and I'll add if I need to.
Yeah, so I think we are a huge fan of validating and doing experiments for everything. So we don't think that
I saw that.
So I don't think the computational prediction is enough, because I don't know whether you have tried it, but when you try to run this epitope R, it gives thousands of peptides, and we add additional filters filtering out only high binding peptides, and there is a possibility that by filtering out the low affinity peptides, we may lose some good quality peptides, and also we also had this criteria of selecting only HLA-DRB1 binding peptide, and there is a likelihood that the DQ peptides can be presented by DP or DQ as well. So I think testing them and validating would help out to find the highest quality peptides. Yes.
I'll add one thing before I know I can go. So, I mean, Paula, you said this before, which I didn't actually know was really interesting. Patients who get 2 kidney transplants sometimes have a lower rate of rejection than one, potentially because of the larger antigen load. And what we think with, if you did every single peptide, maybe that we don't know this, but we wonder if maybe they outcompete each other and then you never get the important epitopes at a high concentration to get the tolerance. We don't know if that's true. It might be fine that you just throw them in there and it works, the system works itself out and it'll work. But they're the things we worry about if we outcompete the important ones to not get tolerance.
And, and then the prediction. So it's a place to start. So if you looked at the prediction of the KD epitopes in the mouse, we predicted 34 peptides, of which 4 were really good. And there may be a couple of others in there that we can, eke out. So it's good at getting candidates. There's a lot of, non-hits. They might even be real binders, but non-immunogenic. But if you look at net MHC PAN 2, it's not as good, which is the Danish prediction algorithm for just in general for MHC2. It's way better than it was the first time, right? So the bigger the data, the learning database for eluted peptides and immunogenic peptides, it's going to get better. It's still going to need to be validated. So we think there's another wave of work to do to use these refined epitope prediction algorithms. The other is, you can use these artificial APCs to potentially give them the whole antigen, elute the peptides, and know the real peptides, right? That's it's a lot of experimental work that's not within our sweet spot of expertise, but it's in somebody's expertise. And so we could make an atlas of DQ peptides presented by DR molecules. That's a doable thing. Yeah. We in the community sense.
Yeah. Also because these experiments are not that simple, and as we saw also from your experiments, when you validate, but the cells 1% of the cell proliferate. So when you actually have to do the assays, it's not that clear cut. I think on the same line, Peter Eager is asking, do the hotspot correspond to the regions of MHC that are the most variable? Which is what I would predict.
Yeah. What's surprising is sometimes, particularly in the mouse, there were polymorphic regions that did not elicit, an immune response, and that probably has to do with whether they had anchor residues that bind, whether they had TCR contact residues that are polymorphic, whether it's a conserved change or a big change. But yes, the short answer is yes, they were polymorphic, but that's not enough.
Yeah. In the human, there's plenty of polymorphic regions which we never really found a response to.
Yeah, but back to that point, like Dr. Kissick was saying, so when we, especially in the human work, when we align the donor and the recipient HLA, there are like 1 or 2 mismatches, polymorphisms. But I think most of especially the region 2 that we think as a hotspot, it constitutes of the many I think like it is the most polymorphic region within this like donor and the recipient HLA. So that's we are the most excited about. Back to the point that Dr. Kissick was saying that we're not like that way we are not going to every single mismatch, but we're just going to one hotspot that is consisted of many like mismatches.
There's Eth Turnquist that is asking if you did align screen your identified hotspots in HLA and see if there's any predicted viral protein sequences that may contribute to heterologous immunity.
Did you do that? I don't think we did.
Not in this project. When in the other project, the CE8 looked for non-human sequences. That's a good thought. We have not done a broad blast for pathogen-specific homologs.
Very easy. We can do that this afternoon.
Okay. Yes, I mean, let him know. There's a question about the mouse experiment where you whether you focused on MHC class, you focused on MHC class 1, whether you have any data on IA antigens, which is the homologue of the HLA-DR, DQ, sorry.
Yeah, so Zhuldyz, looked at the antibody responses. They weren't quite as strong. The skin, this is all a good reductionist system. We're transplanting skin, not heart. There's probably a small number of class 2 expressing cells in mouse skin. I mean, the Langerhans cells express it at high levels. So it would also be interesting to study, but because there was a mouse with KD, because the KD was strongest, it seemed the best model antigen.
And there are a few known epitopes for those, like the IE, and I think Mark has an IA. I forget exactly what they are. We could have compared frequencies. We didn't kind of go into all the different, and we had this, the nice KD expressing mouse that's kind of a, simpler mouse to reduce it down to one antigen. But I think that, no, I don't think any of us think that it's just one antigen on one MHC.
Yeah. And when we do the alignment for this, the class 2 alleles between the donor and the recipients, there are also polymorphism. So there is a likelihood that, by not including it in our tolerance regime, there is a likelihood that we are missing probably good quality antigens as well. But if for the next study, one can look at it for sure.
So I think we have time for another 2 questions. There's again, Neil, that is, there's also something that I noticed. In your mouse experiment, can you explain the bimodal nature of some of the T cell responses, especially in TFH cells? I also remember there were kind of 2 groups of TFH that were positive for.
They are 2 different experiments. I think they are 2 different experiments but both with the same pattern. So the higher group went with the higher control group, and the lower group that was positive was paired. So with a paired analysis, we could have also expressed it that way. But
because I think it's related, like the amount of TFH cells is probably related to the amount of antigen. So if you get, more firm grafts or slightly bigger grafts, but within an experiment, you're pretty consistent. But that's just we had decided if we should put it all together or separate them as supplementaries, but we just thought put all the data in the figure. Yeah.
Okay, thank you. So maybe the last question by John Schmitz. Did you try your peptide pulse and CTLA-4 CTLA-4 blockade without inhibiting CD8 T cells? And if so, what happened?
Yeah,
I think with the
DSA,
there was I think there was an effect on the DSA, but there was no effect on the graft survival. So we think that the by not depleting CD8 T cells, they just the acute rejection mostly comes for the CD8 T cells. That's why there was no effect on the graft survival. But we did see the reduction in the antibody level.
and the same in a valve model. But again, the point here is not to claim that control of the indirect is tolerance to everything. Control of the indirect is specifically a way to prevent the DSA help.
And and that the DSA specifically is contributing to the rejection, because it can be argued that it's just a correlate, but there's no CD8s. The CD4 help is driving DSA, and we're getting rejection from that. We can bring that part of the response down.
And most would accept that in humans, it's likely that DSA, particularly class 2 DSA, contribute to shortened graft survival. It's hard to prove that it's not just a biomarker, but I think the C4D deposition and pathology distinct for antibody-mediated rejection makes it a target.
Okay, I think we hit the hour. I want to thank all for this excellent work and for this discussion, all the attendees for the many questions. I apologize if we were not able to answer all your questions, but I guess you can reach to our speakers. And now I turn over Brian. Thank you again.
Thank you so much. AST would like to thank Dr. Zhanzak, Larsen, Kissick, Cravedi, the leadership of COTS, and all of our attendees for today's session. Please remember to complete the evaluation survey in Zoom and visit myast.org/education to view our video archives and register for upcoming AST Journal Clubs. Thank you all again for today's session.